For chapter 5.6, which is the last part of unit 2 here, we're going to talk about eukaryotic cells, which is us and other parasites, things like that. We'll talk about the structure of the eukaryotic cell and the really important part about it, that it has this interconnection of a bunch of what we call membranous organelles and the endomembrane system. We'll see what that is in a moment. We're going to talk about the functions of some of the parts, the Golgi complex, the endoplasmic reticulum, and the nuclear membrane. Those are all part of the endomembrane system. We're going to talk about the evolutionary process of endosymbiosis, which we've seen previously, but we'll hit again for mitochondria and chloroplasts, which are two eukaryotic organelles. And then we'll talk about motility of eukaryotic cells. Some eukaryotes have a flagella on their cells, and some have hair-like projections called cilia. So we'll see those. Okay, so this is the diagram of a eukaryotic cell. And given this diagram, I'd like you to be able to identify some of the parts of it. The nucleus, which here has a nuclear membrane that surrounds the DNA, the chromosomes of a eukaryote. Eukaryotic chromosomes tend to be linear. Endoplasmic reticulum, this is slightly outside the nuclear membrane and stretches out. This is where proteins get made, and you can see studded on this are little dots. Those are ribosomes. Remember, the ribosomes take mRNA and turn it into protein. So DNA gets converted to mRNA. That goes outside the nucleus to the endoplasmic reticulum, and then it gets made into protein. Those proteins need to go somewhere, so we have the Golgi, which is going to package them up and ship them off. And then we have a couple of other things like lysosomes. These, remember, when things are brought in by endocytosis, they fuse and put in harsh chemicals to degrade whatever's been brought in. Lyse means to break. And then we have the mitochondria, the powerhouse of the cell, right? This is where our chemical reactions that make ATP happen. There's a lot of different shapes for eukaryotic cells, and this is just a small sample of them. In the lab, we're going to look at human cheek cells, so we'll see something from you just like this. But in your body, there's hundreds of different cell types that all have different shapes and arrangements and functions and things like that. Fungi, they can make hyphae. They also make spores many times. Plant cells, here's a plant epidermis. These are stomata, which are little valves that open and close and let gas in and out of the leaf while it does photosynthesis. I did my undergraduate research studying these. They're very cool, near and dear to my heart as well. And this is a protist cell, right? It's a single cell eukaryote that maybe is an amoeba. It's got a bunch of cilia hairs on the outside. It can swim around and find things and engulf them by phagocytosis. So why have all this membrane inside the cell? Well, these internal compartments are super useful because they create little environments where different kinds of chemistry can happen. So in bacteria, they have the cytoplasm, but in here we have all these different compartments where different chemical reactions can occur that can't happen in the cytoplasm. So something like, I don't know, a peroxisome, which is a different type of vesicle, it has a strong hydrogen peroxide inside of it. You don't want that getting anywhere near the rest of the cell, right? That'll degrade the cell, but it can be contained within here, and that peroxisome can fuse with invading bacteria and things like that to destroy them. So these membranes allow different chemical processes and more complex chemistry to occur within the cell. This is really the defining feature of the eukaryote. This system is called the endomembrane system, and that means internal, endo, inside of membrane system. So the membrane system inside the inner membrane compartments. And there's kind of a strict order of things here. We're going to talk about a process called exocytosis first, and then we'll look again at one we've seen before called endocytosis. So exo means leaving or exiting, and cytosis means moving through the cytoplasm. So exiting the cell through the cytoplasm basically is what exocytosis means. This is the process of moving proteins and objects out of the cell. Many things like neurotransmitters and hormones, things like this, need to leave the cell because they're messengers that go outside and talk to other cells. The start of this is at the nucleus. The nucleus has the DNA, and the DNA can get converted into mRNA, right? And then that mRNA goes out to the endoplasmic reticulum, where there's a bunch of ribosomes. These ribosomes take the mRNA and turn it into a protein, and then it gets kind of moved from the production facility, let's say, or the storage facility in the endoplasmic reticulum. It moves out to the shipping system, which is the Golgi apparatus. This is like distribution and shipping. It literally puts kind of like a shipping label on it. It tags the vesicle that it's going to make with a special code that then tells it where to go in the cell. So vesicles with the proteins will bud off from the Golgi and go out to the cell membrane and be released outside the cell. The reverse of this is the process of endocytosis, bringing something into the cell. You might want to bring in a food molecule, say, right? So you might take a large food molecule, do that invagination and create a little vesicle called an endosome. And then this endosome is going to fuse with something called a lysosome, and peroxisomes are a derivation of this. The lysosome has lots of harsh chemicals in it that are going to break down whatever is inside of this endosome. So as they fuse, they release the chemicals, and this all happens in the little vesicle here. So it's not going around the cell destroying important parts of the cell. This helps digest and break down whatever was in here, and then the products get released and the membrane moves back up to the top. This is really critical not only in fighting pathogens, but also in kind of breaking down and recycling components of the cell. The membrane is constantly moving around and around and around, getting moved and reused all the time. So this process of exocytosis, we can see it here actually in a real electron micrograph. It's been falsely colored, but we have, right, the nucleus here. There's little nuclear pores on here that allow mRNA to move out into the endoplasmic reticulum. That's going to then bud off. You can see the green coming up to the Golgi where it gets its shipping label. The Golgi vesicles get released and they can go out to wherever they're going to go. The nucleus is really a very important structure. It has actually a double membrane, and it's really for protection. You don't want things getting to the DNA, right? The DNA is the blueprint of the whole organism. You need that to stay intact. It can't be damaged. Nothing can happen to it. So it's packaged up in a protective shell, and it stores the linear chromosomes of eukaryotes, right? We have 23 different types of linear chromosomes, but we have 2 copies of each. So we have 46 total, right? One from the mother, one from the father. This nuclear pore that we can see here, where's one? Right here, a nuclear pore. This regulates what comes in, what comes out. And we hope that only things we want are going in, but sometimes things like viruses can trick their way in through these nuclear pores and get in here to the nucleus and actually replicate their genome. Remember, viruses can't replicate themselves without using our cellular machinery. They get in here, they replicate, or sometimes they integrate with our DNA. All right, we talked about mitochondria a bit. There's also chloroplasts. These are 2 types of organelles that lead to energy production. And we talked previously about how some people noticed that these things look like bacteria. So they developed this hypothesis called endosymbiosis. Remember, that's inside together living. So we think that an early cell absorbed some sort of bacteria and is now living. It's now living with us, although we've exchanged bits with it quite extensively. So if we develop a hypothesis, we need to present evidence that supports that. That's how science works. So some of the evidence for mitochondria and chloroplast is that they have their own genomes, so they have their own DNA in there, and they have their own ribosomes, very similar to bacteria. Another thing that we see is that they have double membranes, and this is kind of an origami question, right? So this cell is going to engulf the smaller cell. This one has its membrane, and this one has its membrane. So as it engulfs it and surrounds it, the original bacteria is actually going to end up with two separate membranes. And this is what we see in mitochondria and chloroplasts. So this is some good evidence that this actually occurred. So let's start with the mitochondria. They have a double membrane, and this double membrane is really important because this is where our cells do what we call oxidative phosphorylation. We'll talk about that in the next section. This is where ATP gets made, and this double membrane helps that process occur. Remember back to earlier, I talked about bacteria pump hydrogen ions outside the cell, and then they flow back in through ATP synthase to make ATP. Well, that occurs here, but it pumps it into this inner space between the two membranes, and then it flows back in. This structure looks a lot like bacteria. So this is some good evidence that double membrane, the inner one looks like a bacterial membrane, the outer one looks like our cell's membrane. Great evidence. It has its own circular genome. Remember, bacteria had circular genomes. More evidence that endosymbiosis happened. And then finally, that proton motive force turns ATP synthase to generate ATP, just like it does in a bacterial cell. These pieces of evidence, and there's even more that we can present. We can look at the DNA sequence and see that it looks like bacteria. These all point towards us and bacteria forming this partnership however many millions and or billions of years ago. These mitochondria are super critical to the cell, and there are some diseases where we have actually defects in them, some sort of genetic issue. Leigh syndrome is one where oxidative phosphorylation doesn't quite work, and people can be very anemic, or it can be quite deadly. The chloroplast is an organelle that's found in plants. Plants have mitochondria, and they also have these chloroplasts where photosynthesis occurs. The chloroplast has a triple membrane, actually. That's because the original photosynthetic bacteria, they already had a double membrane. So when they got absorbed, they actually have now three membranes. Inside, there's these stacks called grana where little thylakoids occur. They look just like bacterial thylakoids. So they do photosynthesis by capturing light energy and making some ATP. They take that ATP, take CO2 from the air, and turn it into sugars, just like their bacterial relatives. They also have their own circular genome, and this triple membrane points to this endosymbiosis. Now, I've talked about things moving in the cell, right? We talked about the little vesicles get pulled in and they move around the cell. How does that happen? It's probably not something you think about, but it doesn't happen by magic. It actually happens by moving on tracks like a train on a rail. There are a bunch of little fibers inside of the cell that we call the cytoskeleton. So cyto like cytoplasm. This is the skeleton of a cell, and this helps it maintain its shape. It moves things around. I want you to know that there are three different types of molecules in here. They're really easy to remember. We have microfilaments. These are very small ones. These can actually move around. Intermediate filaments, these kind of help stabilize the cell. And then we have microtubules. These are the largest. They're made up of a molecule called tubulin because it makes the microtubules. They're like train tracks that objects can move around in the cell on. And this is interesting because we move things in our cells all the time, but some pathogens exploit this. Herpes viruses use microtubules to travel to the nucleus, and they actually integrate in the nucleus and hide out in the nucleus there. So these are great for us, but pathogens exploit them, of course. All right, finally, some eukaryotic cells need to move around. When we think about single-celled pathogens like Giardia or trypanosomes, things like this, they need to move around. Some of our cells need to move around. Sperm cells and actually other cells in our body have these little hair-like things. So we have two different structures for movement. We have flagella. Usually there's only one, maybe two. These are longer than bacterial in many cases, but there's very few of them. And these flagella, like I said, they don't corkscrew like bacteria, they whip up and down. Then we have cilia, which are hair-like structures, and they're much more numerous. There's many of them, and they beat in coordination. We can see things like amoebas move around using cilia. Both of these need to burn ATP, our cellular energy, to move around. So that's why in the next unit, we're going to talk all about metabolism, which is all about making ATP, which powers life. Okay, that is eukaryotes in a very small nutshell. We have a lot of membranous organelles, right? Things like endoplasmic reticulum, the Golgi, lysosomes. This compartmentalization allows different functions to occur in different places. We have endocytosis, phagocytosis. Those are ways to bring things into the cell. Exocytosis brings things out. The nuclear membrane encloses the chromosomes. It regulates what comes in and what goes out. We have mitochondria and chloroplasts. These are organelles that evolved from endosymbiotic bacteria. I would like you to know some pieces of evidence for why we believe this process happened. The cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. Those act like rails and allow things to move around the cell and also maintain the shape of the cell. And then if a eukaryotic cell needs to move, it could either use flagella. Not all eukaryotic cells have them, though. Or cilia. Again, not all have them. These are powered by burning ATP to move around. Okay, that's it for Chapter 5 and for Unit 2. If you have any questions, be sure to send me an email. Bye for now.